Good morning, friends. Myself, Richa. So today I'm going to talk about three models of DNA replication. So first one is conservative, second is semi-conservative, and third one is dispersive. So there are three models. So Watson and Crick proposed this hypothesis, and Messelson and Stull in the year 1958 they told that the DNA replication might be semi-conservative. So they did an experiment for that. So in this I will tell you about the difference between conservative first, second is semi-conservative, third one is dispersive. So in CSIR NET exam mostly they will ask you about these things. And they will give you this diagrams and you have to depict which one is the correct option. So what will happen after first replication, what will happen after second replication, like this type of questions they will ask. So in the you can see here, this is the parental DNA, which is both double stranded helix DNA, which is blue in color. Now after first round of replication, in the case of conservative one, one new strand will be produced and one old one is conserved here. You can see here the same thing happens in the second replication. One is conserved in both of them and another is produced, right? So this is called as conservative model. Okay, next semi-conservative, in which the parental DNA will separate. You can see here the two strands are getting separated here, and one is old one is new one. The yellow one is the new one. So both of them are getting one old, one new. That's it. So that same thing happens in second replication. You can see one old, one new, one old, one new, and the rest one is one new is produced here in the case of second replication, in the case of semi-conservative model. And in the case of dispersive one, the material in the two parental strands is distributed more or less randomly between the daughter molecules. Okay, so here you can see here. So basically you cannot judge whether the model is semi-conservative or dispersive in case of first replication, but you can judge these things in the case of second replication, whether the model is whether the DNA replication is semi-conservative or dispersive, right? So this is quite a common question what they ask, whether you can depict in the first replication whether it is conservative or semi-conservative or dispersive. So we can judge in the case of first replication, we can judge whether the model behaves like a conservative one, okay, or semi-conservative, but you cannot depict because both are same strands. You can see. So I will show you how the question comes. So wait. Right, so here you can see. So in the case of conservative one, you can see one light band N14 one and one heavy band N15 one only in the case of first replication. But in the case of semi-conservative and dispersive, both are getting the intermediate band N15 and N14. So that's why you cannot judge in the case of first replication what is going to happen. But in the case of second replication, you can see whether the model is following the semi-conservative or dispersive. So what they saw, the most of all the living organisms, they are following this semi-conservative model, where only the N14, the light band, is there, and the higher one, the N15 one, is in the intermediate. N15 and N14, the intermediate one, is here. This is in the case of semi-conservative. But in the case of dispersive, only the intermediate is there. Both are getting the intermediate mod here after second round of replication also. But in the case of conservative one, you are getting N14 and N15 both in the case of first replication as well as in the case of second replication. So that is the difference. So this is very important.